to identify, there's like two proposed at the moment are used. First is to diagnose, and second is for surveillance. For diagnostic or to diagnose the disease are usually really needed by veterinarian to understand what's the bacteria pathogens cause the disease and what type of resistance, so that veterinarian can prescribe proper antibiotics for the farmers. And that's it. If it's done properly, it will reduce the use of antibiotics in animal producers. And it's really proved by in Europe, I think in the Netherlands, in the UK, when they really use this properly, the use of antibiotics in animal reduced drastically over time, because you are really use antibiotics now only for treating the correct disease. And the second one, for surveillance, is really hard to, as I mentioned, because surveillance are, especially when recent bacteria are spreading to the environment. So to identify these pathogens in the soil, in the manure, in the water where the animals are drinking or being served in surrounding environment, it requires more advanced technology, like molecular technology, where you can extract all the DNA directly from the environment. So, and then require more high throughput technology, because you need to monitor thousands of bacteria, not one pathogens anymore, but like many of them at once. So it requires high throughput, because if you measure one by one, it might take a year to even know where is to identify the problem. So there are two technology are currently used. Metagenomics, where you sequence all the DNA that you extracted from these environmental samples. However, antibiotic-resistant genes or these bacteria are usually low abundance. So it requires really deep sequencing technology, and then you need a lot of resources for that, right? Because it's not cheap. Another technology is qPCR, quantitative PCR, which is, I believe, after COVID-19, everyone knows what is technology, right? It's simply quantify the presence of the genes. So this technology is more straightforward, a bit easier and everyone knows also the technology and to understand the data. And the data produced also, it still can be handled by the normal computer.